The fluid mosaic model was introduced by Singer and Nicholson in 1972, and it remains the most accepted explanation of plasma membrane structure. Fluid refers to the lateral movement of components within the membrane, whereas mosaic refers to this kind of patchwork arrangement of proteins and carbohydrates onto phospholipid bilayer. The membrane is selectively permeable, which means it allows some substances to pass while restricting others based on size, charge, and polarity. Small nonpolar molecules such as oxygen and carbon dioxide diffuse directly through the lipid bilayer. Large or charged molecules like glucose or ions require specific transport proteins. Now, let's understand all the components of this model, starting with the phospholipid bilayer. Each phospholipid has a hydrophilic, water-loving head and hydrophobic, water-repelling tails. Because of this amphipathic nature, phospholipids automatically arrange themselves into a bilayer, with heads facing the watery environments inside and outside the cell, and tails hidden on the inside. This creates a stable but flexible barrier. After phospholipids, we have proteins. Proteins are embedded throughout this bilayer. Some proteins lie on the surface, while others span the entire membrane as transmembrane or integral proteins. They assist in transport, communication, and cell recognition. Now, the third component is carbohydrate. Carbohydrate chains attach with proteins to form glycoproteins and with lipids to form glycolipids. These structures help with cell-cell recognition and allow the immune system to identify the body's own cells. And finally, cholesterol sits between the phospholipids, helping control membrane fluidity. It prevents the membrane from becoming too fluid at high temperatures or too rigid at low temperatures, in turn keeping the membrane stable and functional. Now that we understand the components, let's explore some additional key features of the fluid mosaic model. The two layers of the bilayer are asymmetrical, meaning that the outer layer often contains more glycolipids and glycoproteins, while the inner layer has a different composition. This difference is crucial for cell signaling in processes like apoptosis. Also, movement between the two layers is known as flip-flop, which is rare and usually requires specific enzymes such as flipases.